Okay, good morning, good afternoon, good evening. Thank you so much for attending this Metabolomics workshop. Welcome. My name is Sixue Chen. I'm a professor of biology and a faculty director of Proteomics and Spectrometry at the University of Florida. This workshop is part of a collaborative project between my lab and Dr. Sally Assman's lab at Penn State. So I hope you can all hear me and see my screen. So during the workshop, please use the chat function to ask questions. Because we have a big attendance, please mute yourself when you're not speaking. So please do your best or try to use the chat box to ask questions or comment. So our goal of this workshop is to provide an overview of metabolomics technologies and their applications in addressing important scientific questions. This is a Zoom link for both days. And if your colleagues or students who have not received it, please pass along to anyone interested in this workshop. We want this workshop to be accessed by as many people as we can accommodate. As you can see, we designed the workshop. So in the morning time here, we tend to focus on virtual hands-on tutorials. And in the afternoon, we have application examples of metabolomics technologies from leading experts in metabolomics. So I want to thank the National Science Foundation, University of Florida, our ICBR facility, especially Mr. Chris Dillon, who helped us set up the website registration and manage the Zoom, of course, and my collaborator, Dr. Assman, Pennsylvania State University. And I also want to thank Brooker for sponsoring the workshop. Here is Carl Lido and Grace Carr. They are the reps for Brooker. And this is my colleague, Dr. Brian Durham. This is her student L'Oreal, and this is my student Nye. Of course, I want to thank all the instructors for the workshop and you. I also want to thank my lab, especially those who will present hands-on tutorials. They are Yang, Shweta, and Nye. You will see them. So I will just begin to introduce metabolomics. I will tell you why metabolites and metabolomics are very important. I will tell you how we do metabolomics. I will tell you two major approaches, on-target and target. And in the end, I will summarize and show you what we have for this workshop. So let's start with metabolites and metabolomics. I'll just give you an example. Plants is such a big biochemical factory, produce over 200,000 metabolites. And you know, we humans probably don't produce that many. And usually when people look at a human metabolome, usually around 3,000 metabolites. But we humans have a lot of lipid species. So as you know, photosynthesis is very, very important. It uses water and carbon dioxide and make carbohydrates and nutrients for us. And how big is the plant metabolome? Expert Dr. Kazuki Saito published a paper estimate the size of plant metabolome can range from 200,000 to more than 1 million. So this is another example. 2005 Nobel Prize in Medicine was awarded to Yu Tu for her discovery of this anti-malaria therapeutic drug, antimycin. So as you probably know, about a quarter of our drugs are derived from plants. So a lot of drugs we use, they're small molecules and they're metabolites. So what do metabolites look like? And this is a very nice kind of summary of different kind of metabolites, primary metabolites, secondary, lipids, sterols, polyketide, And I just want to show, if you look at this structure, you can see distinct differences from DNA, protein. They have unique kind of building blocks. They have very structured, very kind of building blocks or structures you can easily recognize. But this is to highlight that for metabolites, they really has a big diversity in terms of their structures. That what makes metabolomics pretty challenging and interesting. So they have large range of molecular weight from tens to more than 1,000. They have big differences in their size, a three-dimensional size, and they have different polarity. Some of them really volatile. They have a wide range of concentrations in the cells from picomolar to millimolar level, and they're constantly changing. So every second, every minute, our metabolome is different. And some of them are really unstable, and some of them get degraded. Some of them get modified. So these are just showing you lipids. There are 8 different classes of lipids. And there are many, many different species, as I mentioned. So as you can see, this is a lipid map structures. The curated, more than 20,000, and altogether with computational generated, you have more than 45,000 different structures. And these are a group of specialized metabolites in brassicales. We call them glucosinolates. As you can see, they have really very different side chain structures. Some are aliphatic, some are indolic. This group of metabolites are very, very important for our health. They have anti-carcinogenic activities. So people in Asian countries, they eat a lot of this broccoli cabbage. They have less likelihood of developing cancer. So as you can see, metabolites are very, very important. A lot of us are interested in connecting genome to phenotypes, genotype to phenotypes. So this slide is showing you if you study genomics, and it can tell what may happen. If you look at the transcriptomes, it says what appears to be happening. And if you study proteome, so it will say what makes them happen. And the metabolites and metabolome actually tell you what actually happening or will happen. And as you can see, it's closely related to functions and phenotypes. So where does the metabolite and this terminology metabolome, when did it came from? So it actually came from this paper in 1998. This is also highlight metabolomics is quite young. I mean, has a lot of room for growth. So this slide is showing you sulfur metabolism. Again, I want to highlight glucosinolates, my favorite molecules, metabolites, and their enzymes, myosinases, convert that to isocyanate and volatile compounds, which can help plants fight against pathogens or insects. And as you can see, this glucosinolate pathway is connected to primary metabolism, connect to secondary, this is a secondary metabolism, specialized metabolism. And this also regulated by hormones, salicylic acid and jasmonic acid. And if you look globally, like metabolomics, you're gonna see they have a lot of connections to other pathways as well. So metabolites, they're not only very important in metabolism, but a lot of them are like hormones. They also play important roles in signal transduction. So the signaling network or metabolic network, they're very, very important for us to understand how cells or how organism functions. So I really like this quote. So trying to understand life without knowledge of biochemical networks would be like trying to understand Shakespeare without knowledge of English grammar. So this highlights how important it is to study, to learn metabolite and metabolomics. So next, I will focus on telling you how we do metabolomics. I will begin to introduce mass spectrometry, very basic introduction, and then I will tell you 2 approaches on targeted metabolomics and targeted metabolomics. So, we stand on the scales, we measure our weight, but even the best scale cannot measure the weight of metabolites or molecules. And you can see atomic mass is just so small. So mass spectrometers can really help. So whatever molecules you have, not only metabolites, as long as you can put a charge on the molecules, you will be able to analyze them in mass spectrometers. So for this workshop, we're going to focus on applications or how to do metabolomics mainly using mass spectrometry. So mass spectrometry is actually based on the two equations. One is the Newton's law of motion. So force equals mass times acceleration. And there's another law called Lorentz law. So you have this electrical charge, electrical field, ion velocity, and magnetic field. So if you can figure out this and the acceleration, you put on the molecules, then you can figure out the mass. So this is the basic principle of mass spectrometry. So mass spectrometers are really amazing. So some of them are really big, like 1,000 pounds, 700 pounds in the lab. They're very capable. Some of them are small, and I purchased a 70-pound mass spectrometer like this one here, and there's education outreach website to high schools called moleculardetective.org. So this mass spectrometer actually has been rotating in different high schools. 70 pounds is not very heavy. And there is also a mass spectrometer. It's just a 12 pound, like your laptop, you can carry around. This was built in Graham Cook's lab at Purdue University. In about 5 years or so, you're gonna have an iPhone mass spectrometer. Just to think about what you can do with that iPhone mass spectrometer, right? So we all heard about one apple a day keeps doctor away. And nowadays, if you eat one apple, you can get cancer. How terrible that is, right? So because on our food, our vegetables, our fruits, a lot of times they are pesticides, herbicides, and those are carcinogens. So if you really have this mass spectrometer with you, you can measure how much pesticides or herbicides still left on your food, on your salad, and that really can change your life, right? So it's so important. So these are the basic components of mass spectrometer, and I have a transparent cover here on a small mass spectrometer here. And the first part is ionization chamber. So that's allow molecules to get charges on them, producing ions. So there are many different kinds of ionization. So like electron impact ionization, chemical ionization. I'm going to mention this in a few minutes. And the electrospray and MALDI, these are soft ionization. These allow you to maintain the molecular structure of the ions. And these two, EI and CI, they actually can fragment your molecules during ionization. So I just show you an example, electrospray ionization. So you apply high voltage on the spray needle. So your samples get in from HPLC or direct infusion. So you apply really hot voltage to induce charged droplet creation. And then there's a evaporation process, the charged droplet getting smaller, and a similar charged molecules, they repel, and then they get to really fine single molecule, and they get into the mass spectrometer through the differential voltages at this region. So this is called electrospray. There's also metric assisted laser desorption ionization technology, and Dr. Boone Prentice gonna focus on this tomorrow when he talk about MALDI metabolomics and imaging. So this ESI and MALDI, these are Nobel Prize winning work. So John Fein from Yale University contributed a lot to ESI, and Koichi Kanaka from Japan actually contributed a lot to MALDI. So I will just show you Some mass analyzers we commonly use. For example, this is a quadrupole, you have 4 rods, and this is an ion trap. So these are unit resolution mass analyzers. And so we also have super high resolution mass analyzers, for example, time of flight, Orbitrap, and Fourier transform synchron resonance, ICBR. So I want to show you this mass analyzer here. You can see you have a quadrupole and together with time of flight, and this is actually a hybrid tandem mass spectrometer. So it's very, very powerful. So there are a lot of hybrid mass spectrometers, tandem mass spectrometers we actually use in metabolomics. So I'm just showing you one example with a quadrupole. You have 4 rods. You actually apply alternative AC, alternative current voltages on this rod. You generate radio frequency. So if you apply specific DC voltage to this rod, you can allow one ion to go through at a time. Of course, you can also just use the radio frequency to scan through all the masses through this quadrupole mass analyzer. So I don't have time to explain in detail how it works. I strongly encourage you to watch the 2 YouTube videos I use for my classes. And Dr. Mickey Locks did an excellent job explaining high mass filter, low mass filter, how quadrupole actually resolve separate ions. So as I mentioned, if you have RF, you can separate different ions based on their charge ratio. And if you apply specific DC voltage, you can select different ions. In this case, this blue color ion, of course, they don't have color. So this is for demonstration purposes. So you can select that blue color ion to go through the mass analyzer, while the other ions, they will crash with the rods and disappear. So people actually put these different mass analyzers together, like I showed you a couple slides before, hybrid instrument. So they actually have Q0 for focusing, then Q1, you can do full scan, and you can also do selection. You can select a molecule, ion, and then Q2 is collision cell. So collision-induced dissociation, essentially to fragment the molecular ion here. And CID, CAD is pretty much the same thing, just different vendors, they use different names. Then the fragments will be separated by Q3. So we call these ions the fragments, the product ions, and they get detected. So there are many scan functions for triple quadrupole. First one is full MS scan. You can do it in Q1 or Q3. Essentially, we also call it a surface scan. You look at what molecules in your sample. And then you can also do product ion scan like I just described. You can select a molecular ion and then collide with nitrogen, then get the fragments, product ion in Q3. There are other highly selective and sensitive triple quadrupole scans. For example, precursor ion scan. This allow you just to use Q3 to monitor a specific ion, for example, the minus mode, negative mode 272, and then find out what are the glucosine conjugates in your complex sample, right? So you use this, that's why it's called a precursor ion scan. You use these common fragments to profile specifically the GS conjugates in your sample. And the next one is the neutral loss scan. Neutral loss scan is essentially looking at the mass difference between Q1 and Q3. In this case, I show you example is 162 for glucose. So essentially, whenever you detect the difference between Q1 and Q3 or 162, the mass spec will give you the profiles of all the glycosides. For example, we use this scan actually to selectively profile glucosinolates, which has glucose part of it. And another very specific scan we use very, very often for targeted metabolomics is called multiple reaction monitoring. And I will describe this in about a few minutes. So this is showing you example of full mass scan. I put a lot of words here for you to read later, but you don't have to read it now. So essentially it's a survey scan to look at what molecules are in your sample. And any mass spectra, you can see on the y-axis, you have mass to charge ratio. On the x-axis, you have mass to charge ratio. On the y-axis, you have how many ions hitting the detector. Here is 3.9 E5 hitting the detector. That is almost a million ions, right? So 390,000 ions hitting the detector. And a lot of times people actually make this 100%, so then they use the relative intensity. So this is just an example of full MS scan spectra. So per Doppler scan, as I mentioned, you have a complex mixture at each time. The Q1 will select one ion and then collide with nitrogen gas or helium gas, then breaking them up into fragments. So this is a per Doppler scan. So a lot of times the mass spectrometers are connected with chromatography to have front-end separation. And I'm showing you an example of gas chromatography, GCMS. So you use carrier gas helium and you use high temperature to get molecules off this usually very long capillary column. And then here you have EI and CI fragmentation. So this is harsh fragmentation. So during the fragmentation, the ions actually break up, and then you have mass analyzer to separate the fragments. So here is an example of caffeine, and you can see these are fragments. How you gonna identify this is caffeine? You just search the NIST spectrum database, and you can see the red ones what we acquired, the blue ones are in the library, spectral library, and you can see match perfectly, and you can identify and also quantify based on this intensity of the fragments. But for metabolomics, I'm gonna focus on LCMS here, where front end, you have liquid chromatography. So your mobile phase is actually liquid. And so you have these pumps that allow the liquid to be drawn to the loop where you have auto sampler, you inject samples in the loop, and these solvent will carry the molecules through the loop to your column, inject the molecules on the column, samples on the column, and then the solvent will start eluting, right? Off the column, then electrospray allow us to connect the LC with the mass spectrometer. Then mass spectrometer will separate the ions. So why LC is so popular? It has a wide range of properties of a mobile phase. And you can add different things to your mobile phase. For example, ammonium acetate or ammonium hydroxide, to change the pH. You also have wide choices of stationary phase, which is this column. So you have a normal phase, reverse phase, hydrophilic. You can also modify the functional groups and add different functional groups to these silica or to the C18, for example, the 18 carbon chain to make a specific column. And this then electrospray ionization allow us to connect LC online with LCMS. Also there are many types of mass spectrometers people actually use, but mostly people use tandem or even more mass analyzers, like 3 mass analyzers in one. For example, these tri-braid orbitraps and ion traps allow you to do MSN. So not only just a product scan, but MS3, MS4, MS5 to get down to really fine structures. In general, high resolution mass spectrometers used for untargeted metabolomics. You can get really accurate measurement, and low resolution mass spectrometers, unit resolution. I mentioned triple quadrupole, ion trap. They're usually used for targeted metabolomics. So just an overview of what molecules people can analyze using GCMS or LCMS. You can see GCMS is pretty good for volatiles or anything you can derivatize, make them volatile. And LCMS actually can measure really broad range of molecules, including proteins and large molecules as well. But they have also can cover a wide range of small molecules, specialized metabolites. And GCMS usually molecules with low polarity, and LCMS, you can measure molecules with really high polarity. So how to do metabolomics? Of course, very, very important part is the experimental design and sample preparation. So my colleague, Dr. Kari Basso and Yang Li will give you a tutorial on this subject right after my talk. So now I'm gonna tell you how we do on-target metabolomics. So on-target metabolomics is a discovery, is like a fishing expedition, but a lot of times panel members, grant reviews, they kind of say, oh, you do all this fishing expedition, there's not much merit in your process. But I think they overlooked, this is a discovery. This is very, very important. Fishing is fun, actually. Sometimes you catch a small fish, sometimes you catch big fish, and sometimes you catch really big fish. So this is a really big fish I caught during my sabbatical in Dr. Lloyd Sumner's lab, who is also a speaker in our workshop for tomorrow. So with on-target metabolomics, I already introduced you the LCMS. LC will keep eluting, the molecules will keep being eluted from the LC and get into the mass spectrometer. The mass spectrometer actually measures the total current, total ions hitting the detector. So we call it total ion chromogram, TIC. On the x-axis, you have time. This is a time, and this is a ions hitting the detector, right? And at a different time, just say like this time point, so the mass spec actually do a full MS scan. Remember the surface scan. Look at what molecules in the sample eluting at that time of that point of time, and then they range based on intensity. Then Q1 will select the tallest peak, which is this one, and then collide with nitrogen, break them up, then generate a product ion spectra for this metabolite. And then it will go back and look at the second highest peak, then select that one, do the fragmentation, then generate product ion from that one. Then go back and select the third one. Then just run this like in cycles, right? And then it will, as the metabolites, being eluted from the column, it's just doing these cycles. This way, if the cycle time is really fast, it can capture more and more metabolites. It can go lower and lower in abundance, right? It can, if cycle time is really fast, it can get to most of these metabolites. But you will generate a lot of MS1 spectra, and on these spectra, and these peaks we call it features. And then you will also acquire a lot of product ion spectra. So how are we going to be able to like identify metabolites using the mass spectra we generated? So there are actually different level of identification confidence. Dr. Lloyd Sumner published a paper in 2000 and about a metabolomics standard. And over the years, people have further improved these standards. And currently there are 5 levels. So you can see level 5 is the least confident level, and level 1 is the highest confidence level. So level 5, essentially you have unique features. Those are the M of Z and the retention times. And it really depends on your accuracy. So you see PubMed is a database where there are more than 61 million compounds in there. So if you have unique resolution with a mass of 354, you can see you have 200,000 possibilities, right? So if you use high resolution instrument, high mass accuracy instrument, like Orbitrap or FTICR or TOF, you get down to 1 ppm, you still have 10,000 different possibilities, right? So this is the least confident level. And you can report this level, no problem, is publishable. And then there are level 4. So essentially in this level, you look at isotopic abundance and charge state, R-dot formation, you kind of come up with a formula, okay? But even just with this formula, there are 10,000 asymmetric structures possible, right? So this is the level 4. And level 3, you based on the database search, the libraries, you can potentially identify the tentative structure based on just MS1, which is the the first level of mass spectrometry, right? The feature based on the mass accuracy, retention time, and database matching. You come up with a tentative structure. So this is level 3. And level 2 is another very important jump. So you actually can putatively identify the metabolites based on their fragmentation pattern, based on their MSN data. So this will be a lot more confident, right? Because all the other levels so far, they're based on the MS1. So this is a really good level. A lot of metabolomics publications are based on this level. And then if you really want to validate this MS2 are good, so you have or you acquire authentic standard, and you run that and you validate this with authentic standard. So that's the perfect situation. So that's a level 1. Okay, so I mentioned databases and libraries, and in general, databases are chemical structures generated theoretically or computationally. So for example, Spider is really big database. They have more than 110 million chemical structures on there. And the library usually contains experimentally collected data, like Thermo has this MZ Cloud. They have about 20,000 compounds, more than 8 million spectra in there. So I collaborate with my friend, Dr. Craig Dufresne, and we actually built a spectral library for phytochemicals. So a lot of times different mass analyzers, they generate different kind of fragmentation patterns, and that make a database or library matching really hard. So to overcome this, we actually generate the spectra at a different collision energy. For example, here is collision energy 20. You can see you still have precursor here, and these are fragments. So we also generate spectra like collision energy 30 and also 40. You can see at 40, your precursor has already disappeared, and you generate more intense pattern of the fragments. And you also have other fragments showing up compared to energy of 20. So this is a important way to overcome that variation between different mass analyzers, between different labs. The data usually cannot match really well. And in addition to these different fragmentation energies, we actually also generate different levels of structure information. So this is MS1. We always have MS2 at, we also run positive negative mode. We also generate MSN, as long as we have enough signal. So this is showing you MS3. So this we call spectral tree. So I think that's where future is going in terms of metabolic libraries. So you have the spectral tree, you also have different collision energies, allow people to have higher likelihood to match, to identify the metabolites. So here I just want to list some considerations when you do metabolite identification or chemical structure annotation. It's very challenging, but as I mentioned, all the 5 levels, you can determine those and publish. So one is putative accurate mass identification. And I would definitely suggest using as low as possible based on the intrinsic accuracy. So if you use highly accurate mass analyzers, so you can easily do it, below 5 ppm. But even 5 ppm, there are many different isobaric and isomeric metabolites possible. You also want to consider other formation, like a sodium-nated or modified versions or species, right? So some metabolites can be degraded, some can be carboxylated, some can be hydroxylated. So those all can be considered. Of course, isotopic abundance pattern. So this is an example of alkaloid. So you can see this is where we acquired the spectrum, And this is what it should be theoretically, based on the isotopic abundance of carbon-13, nitrogen-15. So you can see they match really, really well. So this gives you confidence of, this MOZ, this ion is this alkaloid. So something else you want to consider the retention time. But a lot of isomers, they co-elute, for example, citrate, isocitrate, and definitely acquire structure information through MS2 or MSN, MS3. So this is an example I'm showing you. A lot of time we do untargeted metabolomics, we set up some collision energies. They are optimal for most, but not for all metabolites. And for example, this one, you still have a lot of precursor in there. You don't have much fragments at all. But if you're interested in finding out this one, you can always do a targeted MS2. So you can see as you increase collision energy, so your spectral, your fragmentation pattern becomes really rich, right? So this is the way you can acquire high quality MS2 or MSN spectra, allow you to be able to identify the metabolites. So I will give you a couple examples, like how we do this. So using my favorite molecule, glucosinates. So this is a tick I showed you. My spec measures the current, total ion current, and this is a retention time. So let's say my glucosinates is all in there, right? So I will just use 8-MSO as an example. So I do an extract ion chromatogram based on the theoretical peak. And you can see immediately I see one peak here, about 2.2 minutes. So I get retention time. And the peak is so nice, right? So if this peak has only 8-MSO, you can quantify the metabolites based on this peak area. So then in that peak, and we do a survey scan, and we look what is that peak. Actually, mostly you can see is most abundant peak is actually protonated form, right? And we also found another peak, which is sodium-nated, MSOO. You can see this is a sodium-nated mass. So it's so nice they have two precursors. Now I will pick one. Of course, the instrument will select this one for fragmentation. Before that, I want to zoom in this peak to look at the isotopic abundance and see how that match to theoretical abundance. You can see it match perfectly, right? So this is another very strong evidence supporting this is indeed 8-MSOO. And there are no kind of other interfering isotopic peaks in that range. And now we pick that, we do the fragmentation. And before that, of course, you also want to look at mass accuracy, right? Mass accuracy, you calculate that based on the experimental mass, monoisotopic peak, minus the theoretical, divided by the theoretical. You can see this is super accurate, below 1 ppm. And with high resolution instrument, you can easily achieve that. So below 1 ppm and isotope pattern matching, these are all strong evidence. But the most important evidence would be the MSN spectra. So we select this peak, and then we do fragmentation. As you can see, this is a precursor still a little bit left here, and these are the fragments. So the key is to annotate these fragments based on the structure. So you can see in this case, we were able to annotate these two to be fragments of this. And there are some fragments, they could be secondary fragmentation, they could break at different places simultaneously. So it's challenging to figure out what they are. So the software we use in Mass Frontier to really figure out this. Of course, this is really important if your molecule interest is not in any libraries or databases, right? So this is kind of showing you how we identify these kind of molecules. Of course, we also have sodium-nated form. So we fragment that one, and we were able to also annotate these two major peaks from the sodium-nated one. So I hope by this, I showed you how we actually annotate metabolites, and my lab gonna present a tutorial and showing you in real life how we do on-target metabolomics in about an hour or so this morning. So now I'll spend a few minutes just talking to you targeted mass spectrometry, targeted metabolomics. So remember I mentioned triple quadrupole can do this multiple reaction monitoring, and thermal would like to call it single reaction monitoring. Essentially it's the same thing. So you have a complex mixture. Your Q1 will select one molecule at a time, and then fragment. In this case, Q3 would scan only one unique fragment, unique to this, metabolite. And then it will go back, and Q1, you can also select another fragment from this. Usually we use 2 MRM peaks to do the targeted metabolomics. One is for quantification. The other one usually is for verification. So you, of course, you can do 3 fragments, right? But each time you only monitor Q1, a precursor, Q3, a product. So although leucine, isoleucine are isomers, and we cannot differentiate them at MS1 full scan mode, but they can be differentiated based on just on this MRM transitions. Because if you look at their MS2 fragmentation, so these are fragments, you can see these are precursor 132. And if you look at this fragmentation, a lot of fragments are common because these are structural isomers, but isoleucine actually produce a 69 peak, right? So I will show you in a minute that we actually use this to specifically quantify isoleucine. So you actually optimize you have to, for targeted analysis, you know what you're looking for. And for example, you're looking for glucosinolates, or you're looking for amino acid, right? So in this case, I'm showing you an example with amino acid analysis. So there are 21 amino acid, you get all the authentic standards, and you do optimization one by one using the triple quadrupole. For example, glycine, you measure the Q1 and you measure the fragments. One of the fragments is 30, and you optimize the mass spectrometer parameters, including those at the ionization, at the beginning, declustering potential, collision energy, right? And this exit potential at different places. So I just want to highlight, during that optimization, you want to pick unique fragments. For example, I mentioned 69 is unique to isoleucine. You can pick that. And you run standard curve based on your authentic standard. So this is the amount on the column. This should not be the concentration. So you can see we actually do femtomole levels. And this is the examples of different standards you use for your quantification. And then you can run your sample, target these amino acid, and then based on the standard curve you run, you can actually quantify them. So I just want to show you this technology can be very sensitive, and I'm showing you this example from SciEx. You can see they have 1 femtomole column, and nowadays in our lab, we actually can get down to atomole level. So my student, Shweta Chajit, are gonna present a tutorial tomorrow, a virtual hands-on, showing you exactly how we do targeted metabolomics. So these are just some beautiful examples, and showing you the 21 amino acid. You can see they separate really well, and I'm so happy to report that we were even able to resolve leucine, isoleucine on the LC. So you can see their retention time are very different. Their retention times are very different, right? So you have no problem really quantify them. Even the co-elute, as long as you have unique fragments, you have unique transitions. So this Q1, Q3, precursor and fragments, we call this a transition. So as long as this is unique, so even your peaks are co-eluting, you can still be able to quantify them. So these are some other amino acid. And of course, you can also use for proteins that we call mass Western. These are showing you plant hormones. So this is a beautiful, beautiful quantification. You can do relative quantification. You can also do absolute quantification. If you spike in the heavy version of these metabolites in your sample, so you can monitor the heavy version and the light version, you can do absolute quantification using this technology. So this is just to highlight, we have different platforms for metabolomics, different approaches. I just want to show you, no one single platform can cover the entire metabolome. To increase the coverage, the depth, so we want to use hyphenated approaches. We want to do targeted, untargeted, and GCMS, LCMS. So this is showing you nowadays in my lab, we can do more than 800 targeted. We have more than 800 authentic standards. And then we can profile a lot more than a few years ago using untargeted, just because we improved the spectral acquisition, spectral tree, MSN, the different collision energies. So how to report metabolomics data? So this is, again, credit goes to the Metabolomics Standard Initiative. So Dr. Oliver Finn and Lloyd Sumner have been leading the initiative for many years. So these are some recent publications. I'm not going to go through it, but as you know, nowadays, when you acquire this large-scale omics data, the community wants you to deposit the data for metabolomics, like in Metabolite, Metabolomics Workbench, all that. So I just want to list this here for you to read. And then what do we have in this workshop? So we have 2 days, each day about 5.5 hours. And I'm just listing the US Eastern Standard Time, but on the website, you have Beijing time and you have time, London time, European time. So as you can see, right after me will be experiment design and sample preparation. And then we're going to have untargeted metabolomics tutorial. In the afternoon, we have 3 talks showing you how we use metabolomics technologies, targeted and untargeted, to solve real life scientific questions. Very, very important. And then tomorrow, we're going to, in the morning, we have targeted metabolomics tutorials. We're going to have tutorials and applications from Dr. Bon Prentice about imaging, mass spectrometry. And then we have databases and libraries from Dr. Vania, UC Davis. Then in the afternoon, we have leaders in metabolomics, Dr. Oliver Finn and Lloyd Sumner, talking about application examples. With that, I want to thank you for your attention, and I hope we still have a